the weed compete for either nutrients or water. We want to find out how we can actually measure this competition. What information can we get from it which can be useful for weed management? Now, when we are managing our weeds, there are two things which we want to know. It could be more than two, actually. First, we want to find out the weed density, which can actually justify weed control. Then we also want to find out the competitive abilities of these weeds. I know in a given field, we have got a mixture of weeds. But when we control, we just remove all the weeds. But is it that all these weeds are harmful? Or probably there are just a few weeds which are harmful, but we don't know the harmless and the harmful weeds. We may also want to know the competitive ability of the crops. For example, do crops differ in terms of competitive ability? Probably another question is that, are we going to have breeders coming up with crops which can tolerate weeds? So these are some of the issues. So what we are going to discuss will be studies which enable agronomists or breeders to manage weeds. So the first thing we want to talk about will be the population of weeds in the fields, or simply weed density. So we are going to focus on the effect of weed density on crop yield. Now, if we want to find out this information, it means some research has to be done so that we get some information. What we are going to do now will be probably to discuss some of the procedures which are used in research in order to get this information, which is vital for weed management. So there are research designs which have been used to study the effects of weeds on crops. And I've got some of these research designs or procedures, rather. First, we have got what are known as the additive designs. These are very common in weed research. We have got replacement series designs, which are also important in weed research. Now we have got the third. This third, which is known as the addition series or response surface, is actually a combination of additive designs, which is number 1, and the replacement series, which is number 2. We also have something known as the dialer. Now, the dialer can give us some information about the plant individuals. And this information may not be very useful for agronomists and breeders. There's also neighborhood experiments, which may also be useful. But I think for you in crop protection, I will emphasize on 1, 2, and 3, because these are the ones which are used in the field. If you want to find out how the weed is actually affecting the crop and how you can manage that weed. So we will start to discuss or to talk about what we refer to as additive designs. What is the purpose of additive designs? What do they look like? What do we get out of these studies? Are they reliable? Things like that. So first, I will talk about additive experiments. Now, if additive experiments are done, remember we are trying to solve some problems caused by competition, with competition. So if we have to conduct additive experiments, the questions we are interested in or objectives is that number 1, we want to find the minimum weed density which justifies weed control. So there should be a certain amount of weeds in a given field, which necessitates that we should control these weeds. We also want to find out the competitive nature of individual weeds. Although we know in a mixture, usually that is the normal thing in a field. But can we differentiate these weeds according to competitive abilities? Maybe if we can do so and feed this information in weed management, we could do fine-tune the way we control weeds. Now, here I've got an example of an additive experiment. Now, additive experiments, you can actually do them in the actual field, or you can just use pot studies to derive this information. For example, here, this is an example of an additive experiment where we have maize and this weed, Erosin indica, which is competing with maize. I'll talk about the sort of treatments which are used in this kind of a study. Here I've got also an additive design which was used to assess the effects of Tithonia rotundifolia or wild red sunflower in maize. Again, this is a wild red sunflower. You may not see the maize, it's there. And you can see this is actually competing for light in a maize crop. So let us look at the methods Now, in an additive design, it's like you are adding weeds in a field to get certain densities. So the sort of treatments you need are weed density treatments, which you can vary. If it is a pot study, you can vary the density from 0, 2, 4, 6, 8, 10, 12 per pot. But the crop density is held constant. You can see, like in this experiment, where Leucine indica was varied from samples without a Leucine, which is 0, 2 plants per pot, maybe this, I'm not sure. Then 6 and so on, up to 12 plants per pot. But in each pot, you just have one maize plant, which is constant. It also happened here where Titonia, which is the weed, was varied from 0, 2, 4, 6, 8, 10, and so on. So this would be the treatments. Now, because this is an experiment, you want to replicate your treatments maybe 4 or 6 times in RCBD or CRD. This means randomized complete block design or completely randomized design. Now, the sort of data which can be used in this kind of a study is that you collect crop height, crop biomass, leaf area, and weed biomass. And we normally use graphs and regression analysis to understand and interpret the information. But remember, our objective is simply to find the number of weeds per square meter, which justifies weed control. And in order to arrive at that number of weeds, we need to use this kind of experiment. No wonder why our treatments are like this: 0 weeds per square meter or per pot, 2 weeds, 4 weeds, and so on. In this kind of study, normally when you are interpreting information, tables will be difficult to understand. Although some studies have shown some numerous tables in this kind of study. Probably we need graphs. Now, when weed density is related to crop yield or crop biomass, there have been a number of relationships which have been cited in literature. And we have got 5 relationships. Depending on the number of treatments, sometimes if you relate weed density to crop yield, you get a straight line relationship. You get linear relationship, which is represented by this equation. Or you can get, instead of a straight line, when you are drawing these graphs, you get a curve, which is probably a quadratic response. You may get an exponential curve. It could be a hyperbolic curve. Or lastly, it could be a sigmoidal curve. These are some of the responses which you are likely to get if you do such kind of a study in order to just determine the density of widths in a given field, which can give you justification for weed control. I know in cotton, before you can apply a pesticide, you may need to count some insects on a plant, on the cotton plant, which justifies you to spray. So there are certain, what are they? They call them thresholds. For example, if you see number of eggs per leaf or number of larvae per leaf, it may mean you need to spray and so on. Now with the weeds, we have to use a different method in order to arrive at the number of weeds per square meter, which justify weed control. So in effect, we want to look at the effect of weed density on crop yield. Once we understand the kind of relationship which can be any one of these five, depending on where you are. Now, the sort of questions which we wanted to address are as follows. What is the weed density which justifies control of weeds in order to avoid the economic yield loss? Because to control weeds, it can be expensive. So we have to do it when it is really required. The other question is that should we monitor weed populations in a cropping field in order to plan weed control programs? Probably there's a need to go into the fields, make an assessment, and come up with the number of weeds per square meter. Once we have that data, we decide whether to put a chemical or not. The other issue is that is this the best way to predict crop yield loss, to use weed density? Another problem or another question is that normally with these kind of experiments or assessments, you have to use one weed species at a time. So you have to get information for each weed. But if you go in a field where weeds are mixed, is this information reliable? So these are some of the issues. Here I've got an example of a relationship of crop yield, which is related to weed density. I've taken this from Zimbabwe. What it means is that as you increase weed density, at first the response is slow here. the changes are very small. But as you keep on increasing with density, crop yield will start to fall until you are here. If you look at this curve, it's not a straight line. It's like an S. You know, it's curved at the beginning, then it's like straight, then it's again curved at the end. This kind of a curve, we call it a sigmoidal curve. And it was proposed by Zimbabwe here in 1980. Again, here we are trying to show the effect of weed density on crop yield. We are increasing weed density. We are increasing weed density. Here we've got our yield. Sorry. Okay. If you look at the graph, you can see at the start, the changes are small. Then it goes down, and again, it's curved at the end. This is an example of a sigmoidal curve. Here we are just looking at the crop yield. If you are looking at the crop loss due to the presence of weeds, you get something like that. As you increase weed density, in the beginning, there's a small change in the loss. Then there's a big change here. Then at the end, even if you are increasing weed density, the changes are small. What is important is here. Probably at the beginning, it may not matter to control the weeds because the losses are very small. But when it comes to here where the line starts to curve, this could be your weed control threshold. The density which gives you that curve is probably the weed control threshold. Now, on a sigmoidal curve, it is very easy to locate the weed control threshold. So here is our crop yield loss. Here is our weed density. So we can use this model to tell us where can we come in and control the weeds. At a point when we start getting a sharp increase in the loss. Probably you may be interested to read this paper, a simple model relating yield loss to weed density by Roger Cousins. Now, this model was derived using the additive experiment, like the one which I was trying to explain. If I go back a bit, this curve, which is sigmoidal, was proposed by Zimbabwe. And also this kind of crop yield loss, which is a sigmoidal shape, was also proposed by Zimdar. Now, Roger Cousins, who wrote this paper, where he's talking about yield loss and width density, he proposed that instead of getting a sigmoidal curve, the true situation when we have a width density and crop yield, you get this sort of a curve. This is in contrast to Zimdar, who proposed a sigmoidal curve. Cousins' curve is, you start from the top. As long as you have width, your crop is going to suffer. This is what this is being suggested by such a curve. You better not have width. One width, they are enough to cause problems, according to this curve. Now, when Cousins proposed the yield loss, he said it starts from 0, and it's serious when they are width. And it goes up and flattens at the maximum. So he identified 2 important areas on the graph which shows crop yield loss. The first thing which was identified was the maximum yield loss, which is represented by A. And the other thing which was identified on this graph is I. Remember, we are using one weed species in a competition study to come up with this kind of a curve. What it means is that if we carry out this study using various weeds, we can characterize our weeds by these A and I values. And these become indices for competition for various weeds. We can tell whether a weed is highly competitive or not by using these values. That was the important thing about Cousins' study. And you'll find out that it is the common approach which is used to estimate crop yield loss due to weeds. But if you want the weed density, which justifies weed control, according to this, it may mean even if you get one weed, it means you are going to control. It means you have to use control methods. But according to this graph, as a manager, you have to find out the percentage of yield loss which the farm or estate is comfortable with. Is it 5%? Is it 10%? Now, if it is 5%, you have to use this graph to determine the weed density. That weed density becomes your weed control threshold. Probably I will explain this later. What I want to explain again is that this graph, this important graph, can be represented by an equation which was proposed by Cousins in 1985. Let us start from the beginning. YL simply represents yield loss, right? Is divided by this I. And where do we get this I? We get it from here. That is the slope of the curve. So I in that equation is coming from here. dW is the density. So the top part means slope times width density. I dW. The slope of the graph multiplied by the width density. Let me go back to the graph. Width density is here. And it is represented as WD. DW, sorry. Now it's slope multiplied by width density divided by 1 plus everything in brackets. This again, value of slope multiplied by width density divided by maximum yield loss, which is A. So your A here, maximum yield loss, is now in this equation. So this equation defines yield loss. How do we use it? Now, if you have got a certain width density which you think you can accommodate as a farm manager, you have to start with the percentage. Is it 1% which you can, which is not a serious economic loss? Is it 5%? You can just plug the percentage here and substitute YL, then determine the density which you are comfortable with using this equation. Or alternatively, you can just use the graph. Now, here at UZ, we had a student who studied the effect of tithonia in maize. So he varied the density of tithonia in maize. This is the sort of graph which was obtained. So here could be our A, and here could be our I for tithonia. Actually, the values for tithonia for A, it caused maximum yield loss was about 64% in a maize crop. And the slope was 1.4. So we had an experiment where we determined different densities of tithonia, and the calculations came up to this. There was a study which was also done here by Chivinge and Rukuni using Oxaria latifolia density in Brassica napus, which is a horticultural crop. As Oxalis latifolia was increased, the density, the yield started to go down. But if we look at the crop yield loss, according to Cousins, we get this curve, where we have this is the maximum yield loss, and this could be the slope. I think this is the slope for another weed, which was plotted, and we get this sort of curve. So for weed density studies, when you plot yield loss versus weed density, you always come up with this sort of a curve, which is known as a rectangular hyperbola. I should have mentioned it. It's known as the rectangular hyperbola. And you can have the slope here and the maximum yield loss. Okay, let me... Now, this equation, I want you to check that there's one here. Now, in our competition studies, we are assuming that the crop and the weed are emerging at the same time. But in actual fact, you may have a situation where weeds can emerge ahead of the crop. If there's such a situation where weeds are emerging ahead of the crop, then this equation may not be useful. And Cousins realized that he had to alter this one by a certain term. The same equation, but here he substituted 1 with E. E is actually the natural log, which is raised to the power of C times T. C is the rate at which I decreases, or which the slope decreases. But what is more important is the T, which is the time of weed seedling emergence. Once Cousins incorporated this in the equation, then he was able to predict accurately the crop yield loss in the various crops. Now, weed density could be used to estimate yield loss. But there were two scientists, Croft and Spitters, in 1991, suggested that instead of us using weed density to predict crop yield loss caused by weeds, probably it's better to use a relative leaf area of the weeds and the crop. Probably relative leaf area is more accurate in predicting yield loss, and we can use that area to say, can we weed or not? So what they proposed was that they measured the leaf area index of weeds in a field. They also measured the leaf area index of the crop, and they came up with an equation for relative leaf area, which is as follows. You divide the relative leaf area, you divide the leaf area index of the weeds by the sum of the leaf area index of the crop and leaf area index of the wheat. So that ratio gives you relative leaf area. So instead of weed density, they proposed the use of relative leaf area. And this is how relative leaf area is obtained. They also proposed an equation for yield loss. And they said yield loss was equal to Q times relative leaf area. So what is Q? Q is just like I, which is the slope of the curve, hyperbolic curve. And they said the slope could represent the relative damage coefficient. In Cousin's equation, this could be represented by I. So Q times LW, which is relative leaf area, we divide the whole thing by 1 plus everything in brackets. We have got Q divided by A minus 1. And everything is multiplied by relative leaf area. And this, they say, was the best equation to determine yield loss. So instead of using weed density, they proposed relative leaf area, which they calculated as follows. They also proposed that instead of relative leaf area, one can use relative cover. Now, with relative cover, you don't need special things to estimate leaf area. But with this one, you may need to really measure leaf area, and it can be time consuming to do this. That is the drawback for this method. Maybe just counting weeds may be better using the Cousins method. But this one you can estimate whilst in the field. You can say the leaf area, I mean, you can say the area covered by the weed could be 50% or 60% or 80%. Are you seeing the slide?
Yeah, we are.
Yes.
Okay. Yeah, thanks. Right. So so measuring leaf area is a bit problematic, but just estimating relative cover could be probably simple. You go in a field, you say weeds are covering maybe 80% or 50%. You also estimate the cover of the crop, area covered by the crop. So the relative cover becomes your estimated, area covered by the weeds divided by the area covered by weeds plus area covered by crops. And that ratio gives you relative cover. Actually, students have actually found this approach to be easy. You simply go into a field, you move about measuring this relative curve. And the equation now is yield loss becomes, Q, which is the slope times relative curve, all divided by 1 plus Q, which is the slope over maximum yield loss, minus 1, everything in brackets, multiplied by relative curve. That sort of equation will give you the yield loss. So there are 3 ways to do it. It's either you count the weeds. If you are not comfortable with counting weeds, you measure leaf area and try to find the leaf area index of the weeds, leaf area index of the crop. You divide the leaf area index of the weeds plus the sum of leaf area index of the crop and the leaf area index of the weed to get a relative leaf area, which you use in such an equation. Where you use, where there's RC, you put leaf area, where's the, like that. Or you simply go into a field and estimate visually to come up with the relative cover. Once you know the relative cover, you can now use these equations. So this was what was proposed by Croft and now this sort of graph will be similar to Cousins. The only difference is that where Cousins write I, these authors will have a Q. So these are the references which you may read. At this juncture, I will stop. Maybe let me proceed a bit. Here, I just want to highlight the disadvantages of additive designs. Number one, the crop biomass could vary with the species proportion and density. So probably our estimates for weed control thresholds may not be very accurate. Second, the competitiveness of the crop is not known. We may not know the competitiveness of the crop. Another issue is that you are not able to separate the competition of the weeds amongst themselves, because we are increasing density, what we call intra, and the competition between the crop and the weeds, which we call inter-specific competition. You cannot separate this. So it becomes a weakness of this method. But you can actually use it. Let's say you've got different varieties or different genotypes of the same crop. You can use one weed to vary the density and try to estimate the weed control threshold for each variety. Or you can find the A and the I value for each weed across different genotypes. You may find that there are certain genotypes which will alter the competitive ability of the weed. And some genotypes may actually tolerate that weed. So you can use this indirectly, because this may not tell us the competitiveness of the crop, but you can compare different genotypes, using the same weed, and see whether the weed values are changing because of different genotypes. And you can estimate whether a crop genotype is more competitive or not. Here's just Tithonia competing with maize. You can see maize, has been covered. This is Tithonia weed in Murewa. Let me highlight something about weed control thresholds in Zimbabwe. Actually, there is very little research which has been done on weed control thresholds in Zimbabwe. For example, the weeds which we have currently, for example, Elucine indica, Ricardia scabra, which are commonly found, we do not know their competitiveness. We also do not use weed control thresholds for weed management purposes. You can have, the first flush of weeds in summer. Let's say at the onset of rains, we seem to get a lot of weeds, which probably could exceed what is weed control thresholds. And therefore, weed control seems to be necessary every time at the onset of the rains. There's no period where you say, probably I don't have enough weeds. But there are certain conditions where the first flush of weeds is plowed during land preparation. Under those circumstances, the subsequent flushes of weeds in summer may appear to be below weed control thresholds, but studies are really necessary to establish this. Also, in winter cereals, in most cases, the weed seedling emergence appears to be below weed control thresholds because probably it's because of frost and the type of weeds which are now emerging in winter. So you may find that some farmers may not control weeds in winter wheat. But it also depends on the prevalence of frost. So I will stop here for a while